Hi, Hal. Thanks for joining us again today. Your webinar on latest research was very popular.
My pleasure. Lots of great questions, so I'm eager to get started.
Yeah, so we're bringing this all to everybody. A lot of you submitted questions that we just did not have time for. So let's just get started here. So we're just going to start right in the front. Is bicuspid aortic valve disease genetic, and can it be passed to a child?
Yes, so bicuspid aortic valve is quite common in the general population. It's estimated that perhaps 1 to 2% of everybody has a bicuspid aortic valve, meaning the aortic valve only formed with 2 leaflets that open and close instead of the normal 3 leaflets that open and close. Some people with bicuspid aortic valve can also be at risk for enlargement of the aorta, something called ascending aortic aneurysm. Bicuspid valve with or without aneurysm is known to be a genetic condition, but the genetics are very complicated. So there's no easy answer about whether someone with bicuspid valve will or will not pass on that trait to their children, or whether other family members, for example, can be tested. Although bicuspid aortic valve is very common, it's a condition that's not giving up its secrets easily. So despite a lot of work by many groups, we don't know the gene that's involved for the vast majority of people with the condition. I think in people with bicuspid valve, especially if they have an aneurysm, their family members should be screened using an echocardiogram. That would be my best advice. Importantly, in families where some people have bicuspid aortic valve and aneurysm, other people can just have the aneurysm without the bicuspid aortic valve. So if someone has a strong family history of bicuspid valve and aneurysm, and on their first echo, there are no abnormal findings, both the valve are normal and the aorta is normal, it might be a good idea to look again in 4 or 5 years to make sure that enlargement of the aorta hasn't developed. So the answer is yes, it is genetic, but it's complicated and efficient testing is not available. So screening by cardiovascular imaging is the best idea.
Great, thank you. Next, are Marfan, VEDS, and Loeys-Dietz mutually exclusive? Can a person have more than one? Is it possible, or is it likely that a parent has one of these conditions and another family member has a different one?
So in genetics, we've learned that you should never say never. If something can happen, you'll find an example where it did happen. But what I can say is that in a family, for example, with Marfan syndrome, there is no increased risk for another family member to have a different defined genetic condition like Loeys-Dietz syndrome or vascular Ehlers-Danlos syndrome. So if 2 members of the same family had different diagnoses, it would have to be by an extreme coincidence. And therefore, I would say it's highly, highly, highly unlikely.
So does that mean it's probable that one of them did not get the right diagnosis and it's more likely they both have the same one?
Yeah, if one person in the family is being diagnosed with one condition and another person in the family is being diagnosed with a different condition, my first hypothesis, my hunch, is that one of them has the wrong diagnosis.
Okay. Can somebody have Marfan or Loeys-Dietz even if their genetic test did not confirm a diagnosis? So if they have features of one of these conditions and continue treatment as if they had a confirmed diagnosis, is it important to continue to seek the confirmation of that diagnosis?
That's a great question. For Marfan syndrome, current genetic testing is able to identify an underlying gene defect in the FBN1 gene in somewhere around 90 to 95% of individuals. There's still about 5 to 10% of people that by outward appearance have Marfan syndrome by virtue of their physical findings, but where we cannot yet find a gene mutation. Our suspicion is that they also have a mutation in the FBN1 gene, but that it's in an atypical place. It's a place that we're not trained to look. The same is true in Loeys-Dietz syndrome. That's an even more complicated situation. To date, we know that there are 6 different genes that can cause the features of Loeys-Dietz syndrome. But our understanding is that there will be more genes identified. There are quite a few patients that we see that by virtue of their physical features deserve the diagnosis of Loeys-Dietz syndrome, and yet the testing of all the known genes still comes back negative. I would maintain incentive to try to understand the genetic cause of a condition in any family. I think that that can help inform how those folks are monitored and treated over time. There are advances being made in discovery of new genes, and there are also advances being made in improvement of genetic methods. So someone should check back with their geneticist on a regular basis to see if improvements have been made and if new information is available.
Okay, thank you. Would whole exome sequencing show mutations in the genes that you spoke of help to protect people from aortic aneurysm or dissection? And where are these genes located within the chromosomes?
Yeah, so whole exome sequencing is a method to try to find a new disease gene, or to try to find the site of a genetic mutation that causes a condition when the nature of the condition is somewhat unclear or ambiguous, where you're not positive that this is going to be in the Marfan gene or in a Loeys-Dietz gene. So what whole exome sequencing allows you to do is to sequence all 23,000 genes at the same time. The good news is it gives you a lot of information about a lot of genes. The bad news is it gives you a lot of information about a lot of genes that you then have to make sense of. So it's very important when you go into or consider the prospect of whole exome sequencing that your care provider has a strategy in mind to try to sift through the possibilities. If someone has a strong suspicion that there is a specific condition that has an identified gene, it probably makes better sense to simply test that gene or to test a panel of known genes that are associated with aneurysm. Whole exome sequencing is really best reserved for trying to find new genes that cause aneurysm conditions, or trying to solve very puzzling cases where the physical features don't point in any one specific direction.
Okay. Can you please explain sub, splice, and null variants? And is there any research on the null variant in VEDS?
Yeah, so I think it's most important to distinguish between 2 broad classes of mutations. There are some genetic changes that alter a gene and tell the cell to make an abnormal protein. The majority of mutations that cause Marfan syndrome and the majority of mutations that cause vascular Ehlers-Danlos syndrome are in that category. The instructions to the cell are abnormal, but the net result is that the cell will make an abnormal protein. There's another class of mutations that are called null mutations. Other terms include loss of function mutations or haploinsufficiency mutations, but they all essentially mean the same thing. It means that the alteration in the gene essentially tells the cell not to make any protein from that abnormal copy. In some conditions, such as vascular EDS, and in fact, that's the most clear-cut circumstance, haploinsufficiency or null type mutations can be associated with more mild forms of the condition. That can include later onset of physical manifestations. It can even include people that have the genetic change but will never show the manifestations of the condition. In other diseases such as Marfan syndrome, both the types of mutations that cause an abnormal protein to be made and the type of mutation that causes the gene to not give rise to any protein typically cause pretty classic skeletal and cardiovascular manifestations of the condition. In Marfan syndrome, a null mutation or a haploinsufficiency mutation is somewhat less likely to cause lens dislocation, that specific eye feature of Marfan syndrome. But I would say the most important circumstance to try to better understand the mechanism of the mutation would be vascular Ehlers-Danlos syndrome.
In a family with several generations of VEDS, where some lived into their 80s and others died in their 40s, is there a protective gene that enabled some to live longer than others? And I guess same question for Marfan or Loeys-Dietz, when some people in some generations lived longer or shorter lifespans.
Yeah, so the person who posed this question really has identified an issue that is at the center of our current research efforts. So we and others have observed that there can be very wide variation in the severity of Marfan syndrome or Loeys-Dietz syndrome or vascular Ehlers-Danlos syndrome, even within affected family members that all carry the same underlying primary disease mutation. Our hypothesis is that that likely relates to variation in other genes. So we all have about 23,000 genes in every cell of our body. We all carry variants in those other genes. Sometimes those variants are completely silent. They don't have any impact on any physical feature. But other times, those variants in other genes have the ability to modify the consequence of an underlying disease mutation, such as a Marfan mutation. Sometimes those genetic modifiers are protective. They make someone have less severe manifestations. Other times we believe that those modifiers can accentuate the disease manifestations. We're really in our infancy in understanding those modifier genes. But our hope is that if we learned how nature protects some people with Marfan syndrome or vascular EDS or Loeys-Dietz syndrome, we then would be able to mimic that protection using a medication. We can learn nature's secret and then copy nature by using a medication. And that's what our current research is suggesting, at least in animal models. We have been able to identify some modifier genes for both Marfan syndrome and Loeys-Dietz syndrome. And we have learned that we could recreate the protection that was seen with certain genetic variants using drugs. So that's going to be a major emphasis, I think, in the whole field moving forward.
That's exciting. Are there any advances in identifying genes related to FTAD in absence of Marfan, Loeys-Dietz, or VEDS?
Yeah, that's a very active area of investigation. There have been many genes that have been identified for familial forms of thoracic aortic aneurysm or dissection that don't have other associated physical features, so-called syndromic features of disease. And it's very clear that there are many, many more of those genes to identify. So yes, there has been great progress, and there's a lot more work to do.
Is a variant of uncertain significance, or VUS, for fibrillin 2 associated with Beals syndrome? Can it be used to confirm the Beals diagnosis? And is there any research going on now for VUS for fibrillin 2 gene?
Yeah, so I think it's a good time to define some terms. So when we find a variant in a gene, sometimes we're absolutely sure that it's entirely innocent. That might be because hundreds of thousands of people in the general population have been identified with that variant that don't have the condition, for example. Those we can confidently say are innocent or benign variants in the gene. We can dismiss them if we're hoping to identify the cause of a specific condition. Other times we look down at a variant and we're absolutely positive it's the cause of the disease. It's been seen in many people with the disease and not in people without the disease. It's been shown to pass in families along with the condition. So we're really very confident or completely confident that the variant is what we call pathogenic, simply meaning it's the cause. And sometimes, quite often, more times than we all would prefer, we simply don't have enough evidence to make a call. We can't say it's innocent. We can't say it's disease causing. That's called a variant of uncertain significance, or a VUS. Once we identify a variant of uncertain significance, the hope is, the intention is, that more experience will inform the situation. If we keep checking genetic databases and keep checking the medical literature, we'll either gain enough information to say that the variant is definitely innocent or definitely pathogenic. Sometimes animal models can be made with the variant to try to see if it causes a problem, or other cellular assays can be done. But a variant of uncertain significance in the fibrillin 2 gene doesn't tell me a lot. It doesn't tell me that someone has a problem with their fibrillin 2 gene. It doesn't tell me that someone has physical features that would be consistent with an abnormal fibrillin 2 gene, such as people with Beals syndrome, or also called congenital contractural arachnodactyly. So I'd really need to know a lot more information about this individual, their family, the specific variant, before I could hope to weigh in on whether this might be causing a problem or not. I would advise this family to keep in close contact with their geneticist or whoever did the genetic testing, with the hope that more information will become available over time.
Similar question for a VUS in the FBN1 gene, but with a patient that has mild to moderate features. They didn't say Marfan or Loeys-Dietz, but if they have features and a VUS in FBN1, what's your take on that, and what should the patient do?
Yeah, I mean, it's pretty much the same answer. The VUS just simply means there's not enough information to make a definitive call. Again, the individual should be in close contact with their geneticist. And at the end of the day, the doctors caring for this individual or family should be taking care of the physical features that cause the testing to occur in the first place. You know, so if someone has a big aorta, they should be monitored and treated for having a big aorta while people are trying to figure out, is this Marfan syndrome or is it something else?
Right. Okay. I think this from your talk with the 3 Marfan genes that showed promise of causing changes. Are there studies going on about those genes? And do you anticipate that drugs to modify those genes will be more effective than losartan? And we have a lot more coming on losartan in the next section here, but
Okay.
little segue.
So I'm trying to understand the intent of this question. So there are not 3 Marfan genes. We know of 1 Marfan gene, the FBN1 gene. There were 3 different genes that we found that could modify the severity of Marfan syndrome, either in families or in animal models. We were able to show that medications that alter the function of the proteins encoded by these genes were able to influence the severity of disease in animal models of Marfan syndrome. But importantly, those are all medications that would be considered experimental. So they would have to go through the process of being demonstrated to be safe for use in people and then to be tested to see if they're effective in people. It would be a long process before that information could lead to a new therapy that's available for people. In animal models, the efficacy of losartan is pretty amazing. In fact, there's not a lot of room for improvement with very aggressive dosing. But although I clearly believe that ARBs such as losartan have now been demonstrated to be able to afford protection from vascular problems in people with Marfan syndrome, I would also agree that there is room for improvement in the care of people with Marfan syndrome. So the reason for all this work, the reason for ongoing efforts to identify modifiers is to either find something that will complement drugs like losartan or beta blockers, or drugs that are even better, that might ultimately replace current medications. But that's still something that we aspire to. It's not a reality today.
Okay. And one more question in this section. If patients give saliva or blood to researchers in this field, can they get access to the research results?
So it would be very important for folks to talk to the research team. In fact, I encourage them to have this particular conversation at the time that they're being asked to give a sample, rather than after the sample has been given. There should be a consent process when someone is giving a sample for research. And that consent process should outline the steps that will be taken to try to share information with people as it becomes available, with research participants as it becomes available. But in general, I would say most researchers are eager to share information that they believe to be both valid and informative for the participants of a study. But that would require a very dedicated conversation with that particular research team.
Great, thank you. All right. So thank you for the first section of that. And we're going to end this right now. We'll be back with part 2.
Right.
For the wording.
I'm sorry, let me mute the